Okay, everyone. In this explainer, we are going to dissect the critical relationship between cancer and the cell cycle. Our focus will be squarely on the core mechanisms you need to master for the CSIR-UGC NET Life Sciences exam. So, let's get right into it.
Here's how we're going to structure our discussion. First, we'll define cancer as a disease of deregulation. Then, we'll review the normal eukaryotic cell cycle and the importance of its checkpoints. From there, we'll dive into the key molecular players, the cyclins and CDKs. And finally, we will connect these concepts to understand how deregulation of this system leads to uncontrolled proliferation.
So, let's begin with the fundamental concept. At its most basic, cancer is a disease that arises from defects in the very mechanisms that regulate our cells, particularly the mechanisms that control cell division.
Here is the precise definition you must remember. Cancer is characterized by uncontrolled cell proliferation. This occurs as a direct result of defects in fundamental cellular regulatory mechanisms, with the control of the cell cycle being a primary site of failure. To truly understand the pathology, we first need a solid foundation in the normal physiology. So, let's conduct a brief review of the core phases that constitute the eukaryotic cell cycle.
As this diagram illustrates, the cell cycle is a highly ordered sequence of events. It's composed of four main phases: G1, S, G2, and M. A cell can also enter a non-dividing, quiescent state known as G0. However, once a cell commits to division, it proceeds sequentially through these phases, culminating in mitosis.
Let's examine the key events of each phase more closely. G1 is a period of cell growth and preparation for DNA replication. Critically, it contains the restriction point. Once a cell passes this point, it is irreversibly committed to DNA synthesis. The S phase, or synthesis phase, is when DNA replication itself occurs. Following this is G2, where the cell continues to grow and prepares for mitosis, a phase lasting approximately 4 hours. Finally, the M phase, or mitosis, involves chromosome segregation and cytokinesis, and it typically takes about 2 hours to complete.
So how does the cell ensure the fidelity and correct order of these events? It employs a series of sophisticated internal surveillance mechanisms, which we refer to as cell cycle checkpoints.
By definition, a cell cycle checkpoint is a control point where the cycle can be arrested. This pause is not a failure, it's a feature. It allows the cell to verify the completion of critical processes and to repair any DNA damage before it proceeds to the next phase. This is essential for maintaining genomic integrity.
There are several major checkpoints to be aware of. The G1 checkpoint acts as a gatekeeper, arresting the cycle before S phase if DNA damage is detected. During S phase, the S checkpoint monitors the integrity of DNA replication as it happens. The G2 checkpoint is a final quality control step, preventing entry into mitosis if DNA is damaged or replication is incomplete. And finally, the spindle assembly checkpoint halts mitosis if chromosomes are not properly attached to the mitotic spindle, ensuring correct segregation.
Now, what are the actual molecules driving this process and enforcing these checkpoints? This brings us to the core regulators, the molecular engines of the cell cycle: cyclins and cyclin-dependent kinases, or CDKs.
Let's first define cyclins. These are the regulatory subunits. As their name suggests, their concentration oscillates, or cycles, throughout the different phases of the cell cycle. It's crucial to remember that they have no intrinsic enzymatic activity. Their function is solely to bind to and activate their CDK partners. And those partners are the cyclin-dependent kinases, or CDKs. These are the catalytic subunits. They are a family of protein kinases, but their kinase activity is completely dependent on binding to a specific cyclin. When activated, the cyclin-CDK complex phosphorylates target proteins, and this phosphorylation is what drives the cell forward through the cycle.
The specificity of these pairings is essential. Progression through the G1 phase is driven by CDK4 and CDK6 complex with cyclin D. The critical transition from G1 into S phase is mediated by the CDK2/cyclin E complex. Progression through the S phase is then governed by CDK2 partnered with cyclin A. And finally, the transition from G2 into M phase is driven by CDK1 in association with both cyclin A and cyclin B.
Of course, regulation also requires negative control, brakes. This is the role of the CDK inhibitors, or CKIs. They fall into two main families. The INK4 family, which includes P16, specifically binds to and inhibits the G1 CDKs, CDK4 and CDK6. The other family is the CYP/KIP family, including P21. These inhibitors target the CDK2 complexes, thereby controlling entry into S phase and progression through S and G2.
All right, now that we've established the regulatory machinery, let's connect it all directly to cancer. When this tightly regulated system fails, the result is the uncontrolled cell proliferation that is the hallmark of malignancy.
To understand this failure, we must focus on a key substrate of the G1 CDKs, the retinoblastoma protein, or RB. RB is a quintessential tumor suppressor. Its primary function is to act as a brake, specifically to restrict the cell's entry into the S phase.
This leads us to the RBE2F pathway, which is a master control switch for the G1 to S transition. Let's walk through the steps. In a quiescent cell, RB is active and hypophosphorylated. It binds to and inhibits the E2F transcription factors. When the cell receives a mitogenic signal, CDK4/6 cyclin-D complexes hyperphosphorylate RB. This phosphorylation inactivates RB, causing it to release E2F. The newly freed E2F then activates the transcription of genes required for DNA replication, committing the cell to S phase entry.
This quote neatly summarizes how this crucial pathway is subverted in cancer. The anti-proliferative function of RB is lost through multiple mechanisms. These include loss of function mutations in the RB gene itself, amplifications of the genes for its upstream activators like CDK4 or cyclin-D, or loss of function mutations in the genes encoding its protectors, the cyclin-dependent kinase inhibitors.
So, to conclude this section, let's list the principal mechanisms of deregulation. Cancer cells bypass normal cell cycle controls primarily through three routes. First, by overexpressing pro-proliferation factors, such as by amplifying the genes for cyclin-D or CDK4. Second, by inactivating key tumor suppressors, with loss-of-function mutations in the RB gene being the classic example. And third, by inactivating the CDK inhibitors like P16, often through gene deletion or epigenetic silencing.
And this leaves us with a key question that is central to cancer biology. The cell cycle is protected by multiple redundant layers of control: the kinases, the cyclins, the inhibitors, and the checkpoints. The fundamental question then is this: how do cancer cells accumulate the specific alterations required to successfully dismantle this incredibly robust regulatory network?